Hi, my name is Tori Gill, and today I'll be summarizing the phases of mitosis. So when talking about mitosis, first I'll start with the phases of interphase, right? So you have G1, S phase, G2, and then there's also a G0. G0 just basically means a cell that is kind of static or laying dormant. It's not preparing to replicate DNA or create a duplicate identical copy of itself, right? So in G1, basically all the cell is doing is getting nutrients, growing, synthesizing protein, and preparing to replicate its DNA, right? So in the next phase, in S phase or synthesis, the chromosomes will have copies, right? There's a copy of this one, copy of this one, copy of this one, right? So you made copies, and then in G2 is more growth, more synthesis of proteins, organelles, preparing the cell to undergo mitosis, and then late in G2 we'll see centrosomes. Centrosomes are two centrioles each, so if you have two centrioles, it's a centrosome. Not to be confused with a centromere, which can be confusing, but centromere is the center of this sister chromatid. Centrosome, a pair of two centrioles, right? So late in G2, we'll get some centrosomes, right? We'll have our nuclear membrane, duplicated DNA, and our chromosomes, and then we'll be ready to begin prophase. Now, also important to note, in interphase, the chromosomes will never really look like this. You're not going to be able to identify them clearly under a microscope. That's kind of how you know you're in interphase. You may see a nucleus and some just kind of loose, grainy chromosomes, but they won't be easily identifiable like this. And you have your centrosomes. And then you have your nucleus right here, but this will begin to disappear in prophase, and the chromosomes will start to condense, so you'll be able to see them more clearly like this. So then in prometaphase, or late prophase, but prometaphase if you want to break it down further, the centrioles will be at opposite poles, starting to form that mitotic spindle, and the nuclear envelope will be completely broken down, so we'll take out the nucleus. And the chromosomes are fully condensed. You can fully see them. So for metaphase, these mitotic spindles have the microtubules from the mitotic spindle have captured the kinetochores at the centromeres, right? So there's a protein called cohesin at the kinetochore that allows that connection to happen. And the chromosomes will line up along the metaphase plate. Right, so you have your centrioles, mitotic spindle, microtubules branching out, connecting at the respective kinetochores. So once we've passed the spindle checkpoint, all the kinetochores are correctly attached to the microtubules at opposite poles. We'll get into anaphase, where the microtubules pull the chromosomes towards opposite poles, right? Try to get this in. Okay, and as you can see, we have an identical copy on each side, which is what we're looking for in mitosis. So now at telophase, you'll start to see the nucleolus reappearing, preparing to build that nuclear membrane respectively on each side to have its own nuclear membrane. The chromosomes will start to decondense like we saw in prophase, and the spindles will disappear, right? Now at this point, you'll kind of see the cell will have elongated, right? But you'll kind of see this furrow, what's called the cleavage furrow. And basically the membrane is being pulled or kind of twisted like a drawstring. It's being pulled and tightened and prepared for separation. So late in telophase, each new cell will reform its nuclear envelope, and then cytokinesis occurs, which is the actual division of the cytoplasm to form two new cells. Now cytokinesis is complete. You can see we end up with two new cells with a complete set of chromosomes identical to the parent cell. And so mitosis is distinctly for cell replication, right? So you wouldn't see this for sex cells or gametes that require meiosis, sexual reproduction, right, where they need distinctly genetically different daughter cells, haploid cells. These cells are identical to one another. The process is used in like somatic cells in your body, maybe your epithelial lining in your stomach, whatever your body needs to replicate in somatic cells, it will go through mitosis. Now, one difference to note in plant cells, instead of a cleavage furrow forming, what will form is a more rigid cell plate. The cell will have more rigid shape, right, because of that cell wall as opposed to a cell membrane. But similar to cytokinesis in animal cells, how the cleavage furrow in animal cells will turn into respective cell membranes. In the plant cell, this cell plate will become respective cell walls as cytokinesis occurs, and they'll eventually divide into two separate plant cells.